Okay. So as I told you in the first class when we were talking about X-ray imaging, the classical way of producing X-rays is using the vacuum tube. So having a vacuum tube where we accelerate electrons that bump into a metal plate and then send out X-ray as a result. Now, the thing is, this is, as we said before, produces a spectrum, but also because it's in a way a random stochastical process, what happens is that these X-rays are obviously not in phase. They're not like coherent, as we would say. And their intensity is, of course, kind of in a way limited by the technology. Now, we have a second way to produce X-rays nowadays. Well, there's still more, but let's say the other common way to do it, which is particularly for research, and that is using synchrotron radiation. And so what we do there is we take an electron bundle, which we accelerate and which we bend using magnets. And by bending a kind of high energy electron bundle by a magnet, what happens is that the electrons lose energy in the form of photons, and these photons will go tangentially to the orbit of the electrons. So we have several large facilities. So these are actually large research facilities in different countries where we have these large rings, because it's a ring of, say, about 100 meters in diameter or something like that. And so the one here close to Barcelona is this one. Alba is what's called like a light source. So that's the Spanish light source. And that's a synchrotron for research in Spain. And then you have this one is the European one in Grenoble, which is a larger one. It's for the whole of Europe. Now, what can we do with these synchrotrons? So with X-ray, we can do different type of analysis. And so there are several things that we can do. One is we can do, in a way, trying to see what happens when X-rays interact with a surface. So for example, when X-rays fall onto a surface, then, as we said before, X-rays interact with atoms and will release electrons, and we can actually measure these electrons and then see what's going on and then have properties of the surface, for example. Or we can look at X-ray absorption, because what I told you before is that every atom has a specific quantum energy where the outer electron is being kicked out, and it's only for that energy. And depending on the atom, that energy will be different. And so by measuring the energies that are being absorbed and trying to see which energy is coming out, we can actually do kind of a fluoroscopy. And so we can really detect different types of atoms. Next, what we can do is X-ray diffraction. So what I told you before also is that when an X-ray goes through, one thing is that it can kick away an electron, but what also happens is that it interacts with the matrix, and what can actually happen is that the X-ray is slightly bent. So the X-ray continues through the material under a certain angle. And that depends on the whole crystalline, so the 3D distribution of the different atoms within a certain object that we pass through. And by measuring these angles, measuring the scattering, we can actually obtain information on the configuration of the atoms. And then we can do direct imaging. Just very briefly about these applications. So the biggest application is X-ray diffraction. That's being done a lot in these synchrotrons. And that is being done in order to look, for example, at the kind of geometrical structure of proteins. And so what you look at the pharmaceutical industry or in biology, what we have is we have a lot of proteins. It's very easy to determine the atomic kind of composition of proteins. It's very difficult to look at their geometrical configuration within a molecule. And because also one of the things is, of course, we don't have microscopes with enough resolution to directly image molecules and atoms. So we indirectly look at these configurations, or we calculate these configurations by looking at the scattering, X-ray scattering of these kind of proteins, and then recalculate what would be their configuration that causes this. And this is one of the most famous of these X-ray diffraction images, which is actually how they found the double helix kind of structure of DNA, of course, which is very important. So that was done with this type of technique. So then we can look at other ways of diffraction. So you can see, for example, this is looking at some skin, where we can look at different compositions of skin. We're not going to go in detail. We can look at the composition, as we said before, is trying to recognize where, in which structure, which atoms are, and that's especially for kind of heavier metals. And for example, for pollution, you can see in which cellular structures these heavy metals are concentrating. We can look at spectroscopy. For example, here is looking at hair, looking at lipids within the core of hair. So this kind of things can be done with this, but we're not going to go in detail here. It's just if you're interested in some of these things, we can go in more detail. Another thing that you can do is then imaging, as we said before. And so one way to do it is use this for direct imaging and use it, for example, for microscopy. And so there is what we try to do is look at really details of microscopy. And one of the things that we use there is we can actually use the specific properties of X-ray absorption by looking, for example, at the concentration of two different types of atoms. And what we will see is if we look at the absorption spectrum of water versus the absorption spectrum of proteins, so what you do is mainly looking at the absorption of carbon versus the absorption of oxygen, what we will see is that there is a window where we see that carbon absorbs a lot of X-rays, but oxygen doesn't. So that's what we call the water window. So there is an X-ray, which is actually in the soft X-ray, a spectrum where we actually have high absorption through carbon. And water is transparent. So this is obviously ideal to do microscopy or imaging of biological materials, because we know that most of the biomolecules have a lot of carbon. And so we can actually do, in some ways, direct carbon imaging, and that would give us quite some information. So there are microscopes built of this. So these are kind of huge microscopes in these synchrotrons. We're not going to go in detail, but this is like a 40-meter long microscope, for example. And here is some images of this one here in Alba, in the Barcelona synchrotron. We have one of these X-ray microscopes. And one of the things is that, of course, since we work with X-rays, and actually since the soft X-rays, as we said before, the softer the X-rays, the more absorption. So the penetration depth of soft X-rays is not a lot. So we can only image single cells with this. So we have to isolate the cells. And then to make sure that these cells are not evaporated by the X-ray beam, which can be very concentrated, we actually have to work in frozen samples. So we work under cryo conditions. And these are some examples. So the images that you can then make, so you can see like really cell resolution images. So here you see some examples. So you really can look at cellular structures. And actually, you can look at cellular structures. The typical imaging modality that we use to look at cells, of course, or at cell organelles is, of course, electron microscopy. And here you see the comparison. So electron microscopy still has the highest resolution because it can go to much higher energies. So we can accelerate electrons to higher energies than the energy with X-rays that we have. So we can get higher resolutions, but the problem with electron microscopy is that the penetration depth is even less. And we also need to prepare the tissue a lot. So we take a cell, we have to embed it in resin, we have to slice it up, we have to add metals, for example, to stain it. So there's a lot of distortion in the material of electron microscopy, which we don't need to do for X-ray imaging. So we get a little bit less resolution, but we get more like native states, and we can look at full cells. And here are some examples of yeast cells, and here in this case, during the division, that we can image with this kind of X-ray imaging. And then with the proper image processing, we can really get whole cell information, which is very difficult otherwise to get. Okay, of course, for clinical use or to get into information which is clinically relevant. The microscopy is not the most important, it's very important for research. But there's another application actually that we can start using, which we can use at the more like organ level, although we also can get a higher resolution, and which potentially can be integrated in future CT scanners. And that's what's called like phase contrast imaging. So as I told you before, the classical way to do imaging is we have our X-ray source, and we go through the organ and we have a detector, and then we do the projections and have like projections over 180 degrees, get the sinogram, and then do the filtered back projection, and we have our image. Now, this means that we only make use of the absorption of the X-rays. So we let the X-ray pass through, and we just measure how many photons came through from the original ones that we put there. So we just measure what is the overall absorption coefficient. We are not looking at how X-rays are, for example, diffracted. So how X-rays interact with the tissue and how they can slightly change their angle when they pass through tissue. So the result is that with our traditional ways, we can only look at the atomic concentration and not at the atomic configuration. While if we would be able to also measure these angles, then we can, as I said before, look a little bit at the atomic configuration, because the configuration, the 3D spatial distribution of the atoms is what makes that X-rays can bend a little bit. So how can we still try to use this in a more traditional CT way? Well, what we can actually do is if we have X-rays which are all in phase, which is what we call coherent, so which are clean X-rays, what now happens is that if X-rays are slightly bended, so if the X-ray passes through and then changes its angle a little bit, and if I then let them propagate a little bit, what happens is that they, of course, would diverge more and more. So what we will see now is that if we have clean X-rays, what happens is at boundaries of soft tissues, what we will see is that we have more bending of the X-rays than somewhere else. And so if we now place our detector at a certain distance, what we see is that we get what is called a phase contrast effect at the boundaries of tissues. And so this way, we can actually try to look at discriminating different types of tissues. And so there's different setups where we can do this in these synchrotrons. And here are some examples of images that you make with it. Well, first of all, it's being used a lot in, for example, paleontology. So where we have insects, for example, embedded in amber, where the attenuation of the tissue is very, very similar to the attenuation of the surroundings, but by looking at this phase contrast effect, so you see that every boundary here becomes this black and white because of the fact that X-rays are being bended a little bit and then again become either kind of increased in amplitude where they come together and decreased where they have been diverging from each other. This way we can actually make very high resolution images of this kind of setup. Or here's an example of some dinosaurs. This is actually two dinosaurs that were at the same time embedded in stone, where there's relatively little difference in their atomic concentration because of the fact that, of course, if we have preserved kind of tissue, it's like because it's mineralized, so it really became a mineral replacement of the tissue. But by doing this phase contrast, we can non-destructively look at these things. Obviously, when we do medical imaging, we're most interested in soft tissues, in tissues from organs. And here you see an example of where we have brain tissue, which is actually scanned with a CT scanner that makes use of this phase contrast effect. And what I told you before is that a traditional CT scanner cannot discriminate between kind of gray matter and white matter of the brain, but doing phase contrast imaging, you can. And so you see that you can really get very, very high resolution images where we can really discriminate different tissues, even similar to what an MRI can, and actually at higher resolution. Now, because my group is working in cardiac applications, I want to show you some examples of what we can do with heart tissue there. And so we have been scanning some heart tissue. This is actually from another group, but we have similar images where you can really see that now within the heart tissue, you can really look at the structure of the tissue. Instead of being just gray, we can see the structure. And so some of the experiments that we have been doing is, so you take, for example, rat hearts, because at this moment we can only make CT scans of relatively small objects because of the technical difficulties of working in a synchrotron. So what you can do is you can make projections. Here is a typical projection of these, and you can reconstruct it. And what you see now is that within the tissue, within soft tissue, you start to see some contrast. So where you normally don't have contrast in absorption, you do see contrast in phase contrast. And here is an example here is a scanning. This is actually a human fetal heart, where you now see that when you scan through this heart, you see that actually the cells or the fibers are in a very specific way organized. It's actually in a spiral way. We're not going to go in detail now, but this is very important for the efficient working of the heart, that you get the spiral way. Or you can look at what I told you before. When you look, for example, at the aorta here, you see different layers of the aorta, and you actually can see elastic material within the aorta. Now you can then, once you have these images, you can do processing of these images. And here is what one of our researchers has been doing, is trying to calculate the angle of the cells within the muscle tissue. And we know that that is kind of gradually changing from the outside of the tissue to the inside. And you can really see that here nicely. And for example, if you have a fetus that had some kind of cardiac abnormality, you see that this can be very totally distorted. And so you can really measure and interpret this local configuration. Another advantage of this also is because we're looking at improving the contrast between different soft tissues. That means that, of course, also we improve the contrast between vessels, or between blood and vessels and the surrounding. And here is an example of where, without the use of iodine contrast, we can still extract information on the blood vessels. So this, of course, if we can do that in reality, becomes much more interesting, because then we can do, without the need of contrast injections, we could do angiography. And that would be very important, because these contrast agents are potentially toxic, especially for sick patients, especially for kidneys that have to take out this contrast. It could be that there's a problem. So here also, to show you the resolution, we can do imaging at about like 5 micrometer in resolution. Here's an example. This is in the case now with contrast, but it shows you, for example, the high complexity of the vascular structure within a piece of heart muscle. Here is a little bit more detail of an aorta. So you really can zoom in. You see these are actually elastic layers to make sure that the aorta is elastic enough so that we have, with every heartbeat, the aorta can expand in order to efficiently being able to propagate the tissue. So you can prove that if you have an elastic aorta, you need less energy and less pressure in order to get blood through. And then, of course, that's why this is important. And just also to see how this can help in understanding disease. These are actually aortas from mice. This is an aorta from a mouse which has a genetic abnormality that leads to Marfan syndrome. And Marfan syndrome, when we have patients that have this genetic mutation, what we know is that they get problems with the aorta when they're, for example, 30 to 40 years old. They get aortas which are continuously expanding, and which then potentially can rupture or can give dissections. So this is quite dangerous. And you can prove that the reason for this is actually that when these when you have this genetic abnormality, what you see is you get fragmentation of this type of fibers. So these elastic lamina, as we say, they start to go abnormal. And when doing image processing, you can really show that there's holes appearing in these layers, which actually should be smooth, and that this is age dependent. So here you can use X-ray imaging in order to look at details, which otherwise it's very difficult to look in 3D in order to get these things. And you can really look at then discriminating different types of tissues. So here is, for an example, looking at fibrosis. And what you see here is even the resolution of this, the combination of resolution and contrast is good enough that you can see individual cells of heart muscle. Right? So you see here the intercalated discs, and you see all the cells, how they are going together. So here you see that besides using X-ray absorption, which tells us something about the atomic concentration, by looking at X-ray propagation properties, and if we find ways to do that, we can actually get additional information, which is quite useful. And here's an example of also how we can really use this to do histology of 3D kind of tissue blocks without processing, without slicing. And this is an example of a biopsy taken from a patient with a heart transplant, where you can see like infiltration of like white blood cells, and which is like a rejection of a transplanted organ. And then to finish this here, you could do this in real time if you do it properly. And here's an example of an experiment that they did in the synchrotrons. This is done in the Swiss synchrotron that we also collaborate with. This is not done by us, but this is another group that was actually imaging with CT fly, which is flying in real time. And then you can look at doing image analysis. You can really look at all the different internal organs. And this was actually done for studying biomechanics of the flying of the fly. Just to quickly finish, the last thing that we have to cover is, of course, safety. And we know X-rays is a problem. It's ionizing radiation. The dose of X-rays for CT is obviously much higher than for a regular projection X-ray, because we need to do projections from different directions. So that means that actually CTs can become quite high. Nowadays, with a combination of iterative reconstruction, better detectors, better sources, more filtering, we can actually reduce this as much as possible. But it always remains that an X-ray, that a CT gives you, of course, a certain dose. What you then see in these scanners is that really this dose is being measured, because one of the things that you need to remember with X-ray radiation, it's not the radiation at one point which is important, but it's the cumulative radiation that you get during your whole life. So your risk of abnormalities, your risk of cancer is increasing with every additional scan that you get. So it really is cumulative. And that's why nowadays more and more they try to document how much radiation a certain patient gets, and then based on that, of course, try to see or balance the advantage versus the disadvantage of doing imaging and giving the dose. So you really have to see what is the radiation dose that you get. If we can manage to translate what we can do in a synchrotron, so looking at this phase contrast to a CT scanner, and that is currently being investigated in technology research, what you will see is that, of course, if we can look at propagation of X-ray instead of absorption, we, of course, have less biological interaction, because the biggest problem of X-rays is that they are being absorbed and then generate ions there locally, which are highly reactive chemically. But if we only use X-rays to pass through and there's no interaction, then, of course, there's less danger. So we can use like higher energy X-rays with less interaction and less dose. So even by getting additional information on the propagation and then looking at soft tissues and being able to work with less interactions of X-ray, we can actually improve this. So these are things for future CT imaging that I think can make a big, big difference and can actually provide us more information at actually a safer dose.